Namaskar, my dear students. Today we are going to discuss the next topic of the chapter molecular basis of inheritance, and this is one of the most important topic not only of the unit genetics but also of the biology as a whole. Since this method in this method, DNA is the molecule which replicates itself and undergoes duplication. Since DNA is the only molecule which is capable of self duplication, so it is termed as a living molecule. All living beings have the capacity to reproduce because of this characteristic of the DNA. And duplication of DNA takes place in the S phase of the cell cycle. At the time of cell division, it divides into equal parts in the daughter cells. Now, my dear students, DNA replication, so far it is concerned, for the first time, Watson and Crick, who also gave the double helical model of DNA, suggested a simple mechanism of replication of DNA, and on the basis of their double helical structure, they suggested this replication process, and this method was called as the semi-conservative mode of DNA replication. Now it is called semi-conservative because of a purpose. The daughter's DNA actually is a hybrid, conserving one parental polynucleotide chain and forming the second one in accordance to the information got from the first parental polynucleotide chain. That's why, because it conserves one half from the parental portion and it replicates by making one part in the nucleus of the cell, so this is called as semi-conservative. Semi means half. Now it's also called as the autocatalytic mode of DNA because DNA directs the synthesis of DNA itself, because it auto directs the synthesis, itself directs the synthesis, hence it is called as the autocatalytic mode of DNA replication, and thereby it forms the duplicate copy. Therefore, now we have come to know that Watson and Crick suggested a simple mechanism. It is a semi-conservative because half the DNA is conserved. Moreover, it is autocatalytic. If I talk in words of Watson and Crick, they gave a statement in the year 1953, and in their words, I quote, that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material, which was under their notice, and they suggested that this was the way how the DNA could replicate, but actually the mechanism was proposed by Watson and Crick but was discovered by Michelson and Stahel in the organism Escherichia coli, and it was discovered by Taylor, another scientist, in the Vicia faba, and this discovery it occurred in 1858. Dear students, I repeat again that mechanism was proposed by Watson and Crick and discovered by Michelson and Stahel. Now, Michelson and Stahel, they worked on this discovery of this DNA replication by using the radioisotope thymidine. However, Michelson and Stahel, they used radioactive nitrogen. Taylor used radioactive thymidine, whereas Michelson and Stahel, they used a radioactive isotope N15, and when they took out the results after the replication, they found that about the half of the new generated DNA that was containing the radioactive isotope, clearly suggesting that half had come from the first generation and half was synthesized. Now, If we talk about this process, it occurs in a stepwise manner. The DNA replication occurs in a stepwise manner, and the first step of the DNA replication that we are going to discuss is the activation of the dinucleosides. What happens here, that in the cytosol, in the nucleus of the cell and the cytoplasm, the four nucleosides of DNA, that is AMP, GMP, CMP and TMP. What is this? Deoxyamino monophosphate, adenosine monophosphate, deoxyguanine, adenine monophosphate, guanine monophosphate, cytosine monophosphate and thymine monophosphate, because they are DNA, that is deoxy. DE stands for deoxy, and they are freely floating in the nucleus. These are all activated by ATP, as you can see in this figure, that these occur in their monophosphate form, that is adenine monophosphate, guanine monophosphate, cytosine and thymine monophosphate. They are activated by ATP to form deoxynucleosides triphosphates, and then they are called as deoxyadenine triphosphate, guanine triphosphate, cytosine triphosphate and TTP, that is thymine triphosphate. And the enzyme which is required for the occurrence of this reaction is the phosphorylase, and this phosphorylase enzyme, it hastens this reaction, works as an enzyme and acts as a catalyst, and this reaction is called as phosphorylation. Wherever there is addition of phosphates, that reaction is called as phosphorylation. Now these triphosphates, they are in the nucleosides, in fact they are in the activated form. Once they are freely floating in the cytoplasm, they are in the activated form. The next step which occurs is the recognition of the initiation point. Actually, at which place of DNA molecule would replicate, or from where the replication would start, it is initiated at this step, and from a particular point, it can be any point. As you can see here, there is the opening of the double helical structure, because the initiation or the starting point has been recognized, and from a particular point unwinding of DNA molecule starts. That very specific point is called as the initiation point. For identifying the initiation point on the DNA molecule, specific initiator proteins are needed. Now here you must remember that specific initiator proteins are needed, and in viruses and prokaryotes like bacteria, there may be only one region of replication, but in eukaryotes there can be many initiation points, that is at several places the replication of DNA can start simultaneously, which will finally merge with each other, and therefore the initiation point at one point is a continuation which will occur at the rest of the points. After this, once the initiation point for the replication to take place has been recognized, the next step is the unwinding or the unzipping of the DNA molecule. As you can see in this figure, you can see that the DNA molecule, the unwinding and unzipping has occurred. Here you can see just like a zip, it has been represented as it opens. Now what happens in this step? The separation of the two chains of DNA, which is called as the unzipping, it takes place due to the breaking of the hydrogen bond between the two chains. The process of unzipping starts at any particular point, which is called as the initiation point, that we just discussed. That initiation point is specified, and which is then called as the origin of replication. As I told you that in prokaryotes there occurs only one origin of replication. In prokaryotes and in eukaryotes there could be many origins of replication, and at the place of origin of the enzyme responsible for unzipping. There is an enzyme which actually causes the breaking of the hydrogen bonds, and that enzyme is called as the helicase. In this process, unzipping magnesium factor acts as a cofactor. Unzipping takes place in alkaline medium. This you have to remember, that unzipping takes place in the alkaline mediums. Now, single stranded DNA binding protein prevents the reformation of the hydrogen bond, because a question can arise in your mind that once the hydrogen bonds are broken, why don't they attach themselves or remake themselves? There is a protein factor SSB, single stranded DNA binding protein, which stops the hydrogen bond from reformation. And once again, topoisomerase in the prokaryotes, it is also called as the DNA gryase, releases the tension that arises due to supercoiling. As you can see that when the unwinding will take place at one point, for example here the unwinding has taken place, there will be supercoiling here, and this supercoiling, a tension causes on the rest of the DNA, which is released by the topoisomerases. You can imagine it that if we stretch one end of the rubber band, the rest of the end will be stretched. The process of DNA replication actually takes few minutes in prokaryotes, whereas few hours in eukaryotes. For studying it may take a lot of time, but it occurs it is a matter of minutes or two. So next step is the formation of the new chain. Once the replication fork has been formed, then what happens? To start the synthesis of new chain, special type of RNA is required, which is called as the RNA primer. The formation of RNA primer, it takes place with the help of the enzyme polymerase. Now you can see in this figure, unzipping has occurred and a primer is released at the point where the replication fork is formed. Now this formation of RNA primer is catalyzed by the enzyme RNA polymerase, are simply called as the primase. Synthesis of RNA polymerase, it takes RNA primer, it takes place in the 5-T3- direction. What is this 5-3- direction? We have discussed in the structure of the chromosome, DNA. After the formation of new chain, this RNA is removed because it is actually the replication of the DNA, so RNA is not at all needed. Once its purpose is over, it is removed. For the formation of new chain of the nucleotides, these there are obtained in the nucleoplasm. Nucleotides are present in the form of triphosphates like ATP, GDP, CTP and TTP, as I have already discussed. Now after the new chain is formed, there occurs the replication. During replication, two phosphate groups of the nucleotides are separated. In this way, the energy yielded is consumed in DNA replication. So it is clear that DNA does not depend on mitochondria for its energy requirements. It creates its energy by itself from the phosphate bonds. Energetically, replication is an expensive process. The deoxyribonucleoside phosphates thus serve the purpose of, in addition to acting as substrate, they also provide the energy for the polymerization. This is a very important point, that from where the DNA gets its energy, it is not dependent upon the mitochondria. Now you can see in this figure, the formation of new chain always takes place in the 5-3- direction, as it is being indicated in this figure, and as a result, on one chain it will be in the 5-3- direction, and one chain of the DNA will be continuously formed as it is seen in this figure. Now that chain, it will be called as the leading strand. The formation of the second chain begins in the center and not from the terminal point, and small segments are formed, and these segments are made up of small segments which are called as the Okazaki fragments. The discontinuous chain is called as the lagging strand. It will be more clear from this figure. As you can see, on one chain the synthesis is taking place continuously in the 5 3 direction. These zigzag lines are indicating the primers which would ultimately be released of these RNA segments, which are just for starting the replication, and these small segments on the other strand which actually was to be synthesized in the 3 5 direction, but since it does not occur in the 3 5 direction, it occurs in small patches, and because one strand is continuous, that is called as leading strands, other strand is called as the lagging strand. And here the small segments which are seen to be synthesized, these are called as the Okazaki fragments. Ultimately, all these segments also join together to form a complete chain of the DNA. The Okazaki segments, they ultimately join, and they use the enzyme ligase for this purpose. The formation of new chains is catalyzed by the enzyme polymerase. Then actually this formation of the DNA chains, this requires the involvement of some enzymes which are called as the polymerases. In prokaryotes there are three types of polymerases. Just have a look on the different types of the polymerases. First is the DNA polymerase 1. This was discovered by Kornberg in the year 1957. So it's also called as the Kornberg's exonuclease. And what is it is for? It separates RNA primer from DNA and also fills the gaps. And it is also known as the DNA repair enzyme. Second one is the DNA polymerase 2. It is least reactive in replication process, but it is helpful in DNA repairing when there is no DNA 1 polymerase 1 and DNA polymerase 3. When these first and third are absent, the second one, it does the function of the replication, then the repairing of the DNA. Then the third one is the DNA polymerase 3. It is also called as the replicase, and it was discovered by Delucia and Cairns. And large chains are formed by this enzyme, hence this is also called as the replicase enzyme. So these three types of polymerases, very helpful enzymes in DNA replication, they are seen in the prokaryotes. Now some very important points after the new chain has been formed, some important point that we must always remember. First of all, that all the DNA polymerase 1, 2, 3 enzymes, they have 5 3 polymerization activity and 3 5 exonuclease activity. That is clear from the replication process. And if there occurs any failure in the cell division, because I told you in the starting that the DNA replication occurs in the S phase of the cell division, which is intermediate between two actual divisions, and if by chance due to any reason known or unknown the cell division does not occur, this leads to the process of polyploidy, because the replication has occurred but the new cells have not been formed. So now one thing we always call DNAs. Now difference between DNAs and DNAs is that DNAS means so many types of DNA, and DNAASC means the enzyme involved, that is DNAs. So that is the point to be remembered. So this is all about the process of DNA replication. So my dear students, that's all for the process semi-conservative mode of DNA replication. I hope you have understood this process, which is the crux of all the replication, all the cell division and all the reproduction of the world. I hope you will go through this process, and in case of any query, I am available with you on my phone number or on my email for you to leave the message. That's all for today. Thank you.